then continue with the last speaker of this session, and this is Nicole Toasto. She actually obtained her bachelor only very recently from the University of Tampa, and now she's doing her PhD in the lab of Sarah Flanagan at the University of Canterbury in New Zealand. And her research interests include evolutionary and behavioral genetics, and her PhD is focusing around investigating the evolution of sexual dimorphism and mating system variation, of course using Syngnathus as a model system. And today she will talk about her undergraduate work that she did under the supervision of Heather Manson-Jones and Emily Rose. So I'm looking forward to your presentation. Thank you, Nicole.
Thank you so much. I'm so excited to be here talking to all of you. I think I have a video that's going to get played for my presentation.
Yeah, that's Yali's
no worries.
Okay, so please wait a moment.
Good afternoon. My name is Nicole Toasto, and today I'm going to be talking to you about the development of a quantification method for measuring iridescence. And we used the sexually selected traits found in Gulf pipefish as a model system to test this method. So iridescence can be found across a broad distribution of taxa, ranging from ctenophores to cephalopods to various insects, fish, birds, etc. And within these different organisms, the iridescent ornamentation—
I don't mean to interrupt it, but I actually recorded a separate one that I just sent to you earlier this evening that was shorter for my time slot. Since I recognized that it was 15 instead of 20 minutes, I wanted to make sure I only gave a 10 to 11-minute presentation.
So Yalisha sent it like an hour ago or so.
This is the earlier one. I have the earlier one. You sent me this morning?
I sent it, it would have been noon in Beijing.
Okay. Is there any big difference of these two videos?
No, it's just timing.
Okay, maybe Olivia, we can broadcast this one.
Yeah, I mean,
I can also play it myself if that's easier too. I still have it open on my computer.
Okay, you can.
The internet connection seems to be stable, no?
Yeah,
I think so for right now. Yeah.
Okay, so I will stop the sharing and you play your video. Okay.
Awesome. Thank you.
Okay.
Here it is.
Hi, everyone.
Let me pause that. I'm now having issues with Zoom. Here it is. Okay. Sorry about that.
Oh, good. Not your fault at all.
Share computer sound. Perfect. Is everyone able to see the video? Awesome.
My name is Nicole Toasto, and I'm so excited to be able to be here today to share my research with you. This is the project that composed most of my undergraduate, and I'm going to be talking about the development of a quantification method for measuring iridescence, where we used the sexually selected traits found in Gulf pipefish as a model system to test this method. So iridescence, specifically of sexually selected ornaments, can be found in organisms across a broad distribution of taxa, ranging from insects to fish to reptiles to birds And within these different taxa, it has been hypothesized to fulfill a variety of different roles and functions, such as being involved in species recognition or mate choice. Now, in order to determine the function of any ornamentation, it must be measured or quantified in some way. And to date, iridescence has been measured in a variety of different ways. Some of those include looking at the UV reflectance of the wings of butterflies, or quantifying the actual area of color that the iridescent signal is expressed off of, or in terms of presence absence, or the rating on a scale. And so the goal of this study overall was to develop a protocol in which we would be able to obtain quantitative measurements of iridescence. Now, to do this, we needed to choose a model system that would allow us to test whatever method we developed to measure iridescence. And within family Syngnathus, we can see the iridescent ornamentation popping up in multiple different forms, as you can see in the pictures here. And specifically for this project, we used the Gulf pipefish, Syngnathus scovelli. And there's many reasons that we chose to use this pipefish. But one of the main ones is the fact that, as you can see here, the females possess this elaborate iridescent ornamentation on their torsos, and that ornamentation is solely found in the females. And lastly, previous studies have looked into the role of just the physical coloration of these bands and found that it is sexually selected. So it's hypothesized to play some role in mate choice and the overall biology and behavior of these pipefish. So it's an awesome ornament for us to try to quantify. The way in which we quantified the iridescence on pipefish is by the development of a program called the Iridescence Detection and Isolation Algorithm. And this program was built in Access database, and it works in conjunction with ImageJ. And what you do is you upload photos, such as the ones that we have here in the before, and the algorithm processes them, and it basically isolates the iridescent signal of those photos, and it converts it into a false color image, which you can see in the after photo. That's after being processed through the IDIA. So all of that red coloration is what the program is denoting as iridescent. So that then allows us to be able to quantify the signal, because we can just measure the amount of red that's there and get quantitative measurements of iridescence, which is really awesome. So after this program was developed, we wanted to see how it could be used. So I'm going to be talking about two major applications that this program was successfully used in. The first one was a more evolutionary application, where we wanted to see if the IDIA could be used to detect small changes in the variation of this iridescent signal across 4 distinct geographic populations. So photos of female Gulf pipefish were analyzed from 4 locations across the US Gulf Coast, 2 in Texas and 2 in Florida. And you can see those populations outlined here. So I wanted to put these up so that you could see from a visual standpoint, we can tell that the iridescent ornamentation is varying across these different populations. So in general, the photo on top is just the pipefish, the original photo of a female from that location. And the photo underneath is what that same photograph looks like after it's been processed through the IDIA. So some patterns that we can see here just by a visual analysis is that the 2 Texas populations, so the one on top here and the one on the bottom, the top and the bottoms, they seem to possess a lot of post-anal iridescent bands, whereas the 2 Florida populations don't really have that. But in this unique southern Florida location, we see a lot of iridescence on the torso in general that's not constricted to the bands themselves. So those different analyses led us to want to measure iridescent in 3 different ways using this program. And that allows us to look at all of the components of the sexual signal to see if the IDIA is able to detect variation in each component. So the first measurement that we have is something called band iridescence. And this was done by tracing around the red area, the red coloration of every single individual band, as you can see here. All of those measurements were then summed and divided by the total number of bands and reported as a mean iridescence per band. The second way is what we call overall iridescence. And the way in which this was measured was by using the polygon tool in ImageJ to draw a box around the area of interest on the torso of the pipefish. What the program then did, the IDIA, was then able to measure all of the iridescence that was just constricted to within that box on the torso. So this allows us to remove any user bias that could be present from me going in and outlining every band. But this also accounts for iridescence located on the torso, but not on the band. Which leads us to our last measurement. We looked at torso iridescence. So we wanted to specifically identify what iridescence is present on the torso, but not constricted to those defined bands. So what's left over when we remove that total band iridescence from the overall iridescence? So with the IDIA, we were successfully able to detect variation in this iridescent ornamentation, as you can see by the results here. So between our band, our overall, and our torso iridescence, we are able to see different patterns that are arising within each component that the IDIA was able to kind of weed out. So this also highlights the importance of looking at all of the different components of the sexual signal, because they are varying differently based on their geographic location. So they may have different roles in the actual system of the pipefish. But the most important part was that we were able to detect this variation in those different components of the signal by using the IDIA. The second way in which that it was used was a more ecological application, where we were trying to detect morphological changes in male Gulf pipefish now. So no females, we're looking at males now that have been exposed to synthetic estrogen. So previous studies that have looked at the effect of estrogen on male pipefish saw an overall feminization of the male pipefish after exposure to synthetic estrogen. And specifically, what we wanted to focus on was those morphological changes that occur. So you can see here, this is the same pipefish from day 1 to day 22, and a lot of things change about the morphology. But the one that I focused on specifically was the development of those iridescent bands that are normally found just on females. So what we wanted to do with the IDIA was determine if I measured the amount of iridescence using the polygon tool again, the same tool that I used for overall iridescence, on male pipefish at day 1, so prior to any exposure, and then again after on day 18 of exposure to either no estrogen or low levels of estrogen, if I could quantify the estrogen or the iridescence at both these time points and compare them to each other and calculate what we called the change in iridescence, is that going to differ between the control and the estrogen males? And you can see down here, this bottom photograph shows that the IDIA was able to detect iridescent on the male pipefish. And overall, the results show that the pipefish exposed to estrogen had a significantly higher change in iridescence than those that were not. So the IDIA can be successfully used to detect these morphological changes. And most importantly, this may be able to be applied to a wild population. So if we generate some standard curve between estrogen amount and the level of iridescence, we can then use male Gulf pipefish as indicators to the amount of contaminants such as estrogen in wild aquatic populations. So overall, the iridescence detection and isolation algorithm, the IDIA, was successfully used to detect minute variations in band, overall, and torso iridescence across 4 locations. And it was also able to detect a change in the development of iridescence in a male scovelli as a result of exposure to synthetic estrogen. Future directions for this program are limitless, but they're mainly centered around the continuation of the use of this IDIA, this program, for Gulf pipefish to determine things like its role in their mating system, the plasticity, what environmental factors are influencing it, but also the modification of the parameters this program uses so that it can be used for either other Syngnathus or other organisms in general. And by having those quantitative measurements, we can finally decipher the role that iridescence plays across the animal kingdom. So I just wanted to say thank you to everyone who helped me with this project, especially my co-authors and my mentors, Emily, Sarah, and Heather. And with that, I will take any questions. Thank you.
Thank you very much, Nicole, for this very nice presentation. I see that Nuno has a question.
Yes.
So this is the inescapable question. Where and how can I access your algorithm to test it?
So that's actually one of the future studies is we want to move it from being built into Access database to a more publicly accessible, so something like GitHub, where we can upload the code for this program into there, but that has not occurred yet. It is hopefully soon to be happening. So.
Okay, thank you. Very cool, your research. Thank you.
Thank you.
Yeah, really nice. We're all looking forward to be able to use the tool. So, questions?
What?
Not a question. So, you said that you have exposed them to then synthetic estrogen, and then it has changed the iridescence of the males as well. So, did it also change their mate choice behavior, or you only kept them individually?
So the photos that I used were actually from a study that was done by Dr. Rose during her PhD, and she just kind of sent them my way so that I could use them in this analysis. But they found that after exposure to estrogen, they also have difficulty reproducing. So the males have a hard time either getting pregnant or carrying those eggs to gestation. So there's definitely behavior that could be influenced from exposure to estrogen as well. We just focused on obviously the morphology changes for the purposes of this study.
Thank you. Very nice.
Thank you.